Thank you all for being here. As you've heard already, my name is Andrea. I'm doing my PhD in the Neural Control of Movement lab at ETH Zurich. So, in our laboratory, we are mainly dealing with movement-related matters of the brain. We are interested in investigating, for instance, how the brain acquires and controls new movements. We are also interested in investigating how the brain encodes the movements of other people, how it recovers after injury or after degeneration. But we're also interested in building really cool devices with which we're able to modulate the function of the brain. And many of the questions that we ask in our lab can be answered through various forms of brain stimulation. This is why in my talk I want to present you two of these techniques and show you how we can use this to modulate or to read, basically, the changes that happen in the human motor system. Let's see if this works. Yes, it does. So, for most of us, hearing the words brain together with stimulation creates the sort of image that makes us think that we need to inject electricity directly to the brain. And actually, we wouldn't be really wrong, because one of the main or most commonly used forms of brain stimulation is that stimulation through electric fields. And you can see here the first demonstration of this technique happening in 1980s at University of Cambridge. What they were doing here is they were stimulating the right side of the brain of this participant. They were mainly focusing on the human motor region. So it's basically the brain region that controls all our muscles in the body. They were stimulating specifically the part that controls the hand muscles, and as you can see here, the participant is directing his attention to his left hand, which will actually twitch when stimulation will be given. So for the primary motor cortex, this is the setup that they use, with an anode or positive electrode over the area of interest, in this case over the motor area, and the cathode or negative electrode over a few centimeters in front. And the first paper presenting this technique actually mentioned that they need to apply brief but very high voltage shocks to the brain in order to obtain a response. And actually, it's possible to stimulate the human brain without undue discomfort. And if you look at the voltage here, some of you may know this is a very high intensity. But if you don't know, just to give you an intuitive understanding, a normal battery that we would use for our flashlight, for instance, has like 1.5 volts. The car battery has like 12, and the normal power supply that we have in every room has like 230 volts. So applying 2000 volts to the brain is quite a high intensity. But still, they say in their paper that this is that they can stimulate the brain without undue discomfort. And that's because this technique was actually quite revolutionary. Before the introduction of this technique, the only way to stimulate the brain and to obtain a response in the muscle was by cutting the skull open and sticking some electrodes to the brain. So of course, this is quite nice. However, applying 2000 volts to the brain is quite painful, and actually this technique was not used too much for clinical or research applications. But now, what if I were to tell you that I can also stimulate the brain by simply placing a magnet close to it? You may think that I'm kidding. But actually, nowadays we can penetrate the skull, stimulate the brain, and elicit the same muscle responses simply by placing a magnet close to the participant's head. The idea, you can see this technique demonstrated in this picture. It was introduced in 1985, and how it works is by simply placing a coil through which an electrical pulse is sent near the participant's head, and this would elicit the same muscle twitch. So you can see a similar thing. Here, the right human motor cortex is stimulated, and they would observe a muscle twitch in the contralateral side, so in the left hand muscles. This technique is called transcranial magnetic stimulation, and as I said, it was introduced in 1985. But actually, magnetic stimulation has been around for quite some time before. Here you can see a demonstration showing the use of magnetic stimulation back in 1892. They had a similar technique, so a coil through which an electric pulse was sent, and this was set somewhere around the participant's brain. Of course, this guy wouldn't be holding it during the stimulation. Other people have tried to be a bit more focal, so they apply two coils on the right and left side of the brain and try to stimulate it, or more extreme cases in which the participants had to be completely immersed in this magnetic coils. However, the technique that we use today is quite similar to this, so it's a modern, non-invasive and painless stimulation. We don't need to cut the skull open, and we don't need to apply any painful electrical pulses to the brain. What is actually more remarkable and unusual about the introduction of transcranial magnetic stimulation is that its application in humans and also in patients even preceded any animal experiments which would look at what are the mechanisms of this technique. So they went directly to humans and to even patient studies. So maybe it seems a bit trivial now, stimulating the brain and obtaining a response in the muscle, but actually this information can tell us a lot about the state of the brain, about how excitable the brain is, about how healthy it is, about the state of the corticospinal tract, and many other things. And this is why these techniques are used a lot in clinics. So, how does this technique actually work? Well, in 1831, Faraday made a discovery that forms the basis of transcranial magnetic stimulation. His discovery showed that a time-varying magnetic field can be used to introduce an electrical current. So the whole idea is that TMS works based on electromagnetic induction. And I mentioned time-varying magnetic field, and this is quite important because if we place participants in a static magnetic field, although very strong, like in the case of magnetic resonance imaging, which actually gives us this nice pictures of the brain, nothing happens, so we don't stimulate any nervous tissue. However, when we vary this magnetic field, we can obtain a secondary electrical current in a nearby conductor. So, for TMS, we use a coil, which is usually actually formed by many copper wires. We send an electrical pulse through this. This electrical pulse will generate a magnetic field around the coil, and as I've told you, this magnetic field will generate a secondary electric field in the brain. And although the mechanisms are quite complex, we can just assume that this sort of stimulation will lead to excitation of neurons. And when we stimulate the human motor system, this sort of impulse that we elicit will travel to the spinal cord and to the muscle, and it will elicit a muscle twitch. So I'll show you this again. It's quite cool. So, by stimulating the primary motor cortex, we can obtain a direct motor output. But what do we really activate? Well, the cool thing about stimulating the primary motor cortex is that this region contains a map of the different muscles in our body. So this is called the motor homunculus. You cannot see it really clearly here, but this is basically a map that shows the correspondence between which brain area controls which muscles in the body. And you can see here, for instance, the toes, they are represented somewhere in between the two hemispheres of the brain. Then we have leg muscles, trunk, shoulder, arm, elbows, wrist, and so on. And the interesting part about this map is that the size of the cortical representation of this muscle is not a function of how big the muscles are, but more about what sort of actions they perform. So for instance, the hand muscle has a very big representation because we use these muscles a lot to perform really precise movements. So we need a lot of brain area to be able to do this. And as I showed you before, we stimulate this. So for instance, we could actually stimulate hand muscles. And this is what we do a lot because this region is quite outside of the brain. We can stimulate it easily. It's also a big region. And this is what I will be talking in the next slides. So with this technique, we can stimulate quite small brain regions, around 2 centimeters, but we cannot penetrate really deeply in the brain. But I will show you now what we actually record in the muscle. So we stimulate the primary motor cortex and record responses. I call them muscle twitches until now, but actually they are called motor evoked potentials. So this will be motor responses that are produced by stimulating the primary motor cortex, and they are normally recorded by using surface electrodes. So we just stick some electrodes over the muscles, and we are able to record responses that look something like this. And we can look at many aspects or many measurements related to this motor evoked potentials. But what I do in my research is I analyze the amplitude of the motor evoked potentials. And this amplitude is actually a proxy for the level of excitability in the primary motor cortex. So what do we see? What are these amplitudes telling us? Well, for instance, when we record, when we stimulate participants that are at complete rest, we can record motor evoked potentials, and we could compare them with the case when participants are preparing to perform a movement. And here we can see that the amplitude is already higher. And this amplitude is even higher when we actually execute a movement. But what I do in my research and what I think is more interesting is that even when participants are completely at rest, we stimulate their brain and we just show them a movement. So their hand muscles are completely relaxed and we show them the hand of someone performing a grasping movement, let's say. Or they can even just think of a movement, and this will actually also modulate their motor evoked potentials. And I'm doing my PhD using this technique, but I also use it during I used it during my master thesis, and I really, really liked it because we can find out so many things about the brain. So, for instance, in my research, I'm interested in what happens in our brain when we observe the movements of other people, but also what factors influence this modulation of the motor evoked potentials. How can we make these responses stronger? Why does the brain care more about some movements and not others? Or also, we can find what sort of neurons are responding to these movements. And I mentioned grasping movements and fingers, and this is what I usually show to my participants. I show them videos of a hand reaching, grasping, and lifting the sort of cylinder, either using a whole hand grasp or a precision grasp. So in the whole hand, we use all of our hand muscles to perform a precision grasp, just 2 fingers. And I stimulate the primary motor cortex, the hand motor area, and record responses from 2 different muscles, pinky or the little finger or index finger. And I record the responses from here because these ones are particularly involved in performing these 2 actions. And this is what we can see also in the recorded motor responses. So we see pinky finger shows increased amplitude during the observation of whole hand grasps, while index finger shows increased amplitude during the observation of precision grasp. So although participants are just at rest, by looking at the pattern of motor evoked potential, we can see basically what sort of movement they are observing. So I showed in my experiments these participants this sort of movements, but I was interested in finding whether they really need to see all the aspects of the movement. Is it really necessary that they see this hand opening and grasping the object, or I can just tell them what's happening and their brain will answer in the same manner? So this is why I show participants videos that are preceded by some colors. So they learn before the experiment that white cues will precede, for instance, precision grasp. And I will show you now a video of how this works. So they see a white cue, and now they see this hand reaching and grasping with the precision grasp. And they also learn that blue cue precedes whole hand grasp. And I show them these videos like this, full view, but I sometimes also occlude them. So I cover the most important part of the action. There is no way that participants would know whether there is a whole hand grip or a precision grip. And I will show you now what I find. You can see here these videos. And what I do actually with transcranial magnetic stimulation is I stimulate at different time points, either during the reaching part. Here you can see that the hand actually is in a similar position in both movement types, so it's just a clenched fist. It's before opening to grasp the cylinder. I also stimulate them a bit later when they're just when the hand just grasps the cylinder, and even later during the lifting phase. And what we found is that the pattern of motor evoked potential is predictive of what movement is being shown during the grasping phase, because of course participants can see clearly what is happening. During the lifting phase, of course, the same, participants can really see this movement. But what was interesting is that even during the reach phase, when there is just a clenched fist, the pattern of motor evoked potentials was predictive of the movement that was about to start in this case. So this indicates really that the colors that we showed them before and that participants learned to associate with the movements were enough to modulate the motor evoked potentials. But now, what's even cooler is that when we even occlude the movement, of course this is just the same, so we observe the same responses here, but even when participants don't see any movement, if we stimulate them, the pattern of motor evoked potentials is still predictive of the observed grasp or the predicted grasp type. And this modulation is not even, it doesn't just last for a few seconds, but even later we see the same pattern. So even without seeing any movement, participants were still, or the primary motor cortex of participants was still predicting the specific grasp type. So we showed that we don't need to see movement kinematics, that it's not really, that the primary motor cortex doesn't need to see the whole movement when it has some contextual cues or some prior information about what is going on. So I like to think that this project showed that even if you hide, my primary motor cortex will know what you're doing, because it can combine different sources of information, it can take context into account and also observe movements. So I would like to conclude by thanking you all, my laboratory and also the REACH community, but I will show you this last slide and I'll just leave it here with the clinical applications of these techniques, which are many, and it's not my expertise, but I'll be happy to talk more about it.